Hello viewers, this is Dr. P. Mary Anupama from the Department of Biochemistry, St. Joseph's College for Women, Visakhapatnam. Today, let us study the topic enzyme specificity and active site. The contents of the presentation include definition of enzyme specificity, then we shall see the different types of specificity, and then about the active site. We know that enzymes, they are very much specific in their action. They specifically bind with the substrate, which we can say that it is substrate specificity, and the reaction that they catalyze, that is also very much specific. So they exhibit substrate specificity as well as specificity with respect to their reaction. Let us see some examples. Say for example, we have hexokinase. Hexokinase, it phosphorylates all the hexoses, but it does not phosphorylate the disaccharides or the trisaccharides or any other molecule. So here these hexokinases, they phosphorylate hexoses giving rise to the respective hexose 6 phosphate, and it is a broader specificity, while we have glucose 6 phosphate formation which is catalyzed by a specific enzyme glucose kinase. Here the glucokinase, it phosphorylates only glucose and ATP is involved in this reaction. Let us now study about types of enzyme specificity. We have three types of enzyme specificity, that is stereospecificity, then we have reaction specificity, and we have substrate specificity. Stereospecificity, there we'll be studying about the three-dimensional conformation. Reaction specificity is with respect to the reaction in which they catalyze, while substrate specificity, there we'll be talking that they specifically bind with a particular substrate. Stereospecificity. The enzymes, they can differentiate between two stereochemical forms which are varying with respect to their three-dimensional conformation, and thereby the enzymes exhibit specificity. Say for example, this is one form of a molecule, while this is the other form of a molecule, which are varying with respect to their three-dimensional conformation. And the best examples are the L-amino acid oxidase and the D-amino acid oxidases. L-amino acid oxidases, they specifically act on L-amino acids, while the D-amino acid oxidases, they act on D-amino acids. So thereby they are exhibiting that stereospecificity. Some enzymes, they result in the formation of stereospecific products. For example, in the reaction catalyzed by glycerol kinase, which phosphorylates glycerol, and it results in L-glycerol 3-phosphate. Initially, there is no asymmetric carbon in the glycerol, but by the end of the reaction, we see that it is L-glycerol 3-phosphate which is formed. Thereby, these enzymes, they are resulting in the formation of stereospecific products. Reaction specificity. In this, we have a particular substrate which can undergo different reactions with different enzymes. And these reactions that it undergoes with each of the enzyme, it is very much specific. For example, we can see this in case of amino acids, where each amino acid can undergo deamination, decarboxylation, transamination, racemization reactions with different enzymes. Say for example, glutamate, it can undergo oxidative deamination, it can undergo decarboxylation as well as transamination with three different enzymes. Substrate specificity. Here the enzymes, they are very much specific in selecting a particular substrate. They act only on that particular substrate. We have several examples for this. Let us see two of them, of which the first one is the reaction catalyzed by glucokinase. Glucokinase, it specifically phosphorylates glucose, resulting in glucose 6-phosphate. And one more example that we have is urease. Urease is the enzyme which is involved in cleaving urea, and it gives rise to ammonia and carbon dioxide. So here both these enzymes, they are very much specific in their action. Now let us study about the active site of enzymes. This is proposed by Alexander Oxton in the year 1948. According to him, he says that there are three different points of interaction between enzyme and substrate. So here, the active site points, they are represented by A1 to A3. Here the enzyme, it has this active site, and then these are the points of interaction of the substrate from R1 to R3, whose orientation will enable specific binding at the active site of the enzyme with these groups. So this particular active site, it is contributed by very few amino acids. The enzyme is a protein having many amino acids, but this catalytic site and that site which is having this particular active sites, it is contributed by very few amino acids, while rest of the amino acids, they contribute to the structure and shape and size of the enzymes. So this interaction will enable strong binding between enzyme and the substrate, resulting in enzyme substrate complex formation, and thereby the substrate is later on converted into product. Later on, with the advent of X-ray crystallographic studies, the three-dimensional structure of proteins and the enzymes was elucidated. These studies, they have revealed the active sites of the enzymes. They identified them as small clefts or pockets which can accommodate the substrate. And here we can see the X-ray crystallographic structure of chymotrypsin, and this is the active site accommodating the particular substrate. So it is involving only few amino acids. Not every amino acid of the enzyme is involved in the formation of this active site. Now let us summarize what we have studied. We were studying about enzyme specificity. There are three types of specificity: stereospecificity, reaction specificity, and then substrate specificity. And then we have studied about active site, because specific binding is because of specific interactions between the enzyme and the substrate at the active site. We have seen that the amino acids at the active site, they are few, but they contribute to the binding of the enzyme and the substrate. The active site is a small cleft. It is a small pocket of the enzyme which accommodates the substrate, leading to the formation of enzyme substrate complex. Thank you.